All right, good evening, everyone. We'll go ahead and start on time. Thank you so much for coming this evening. My name is Andersen. I'm director of the Stem Cell Research Center here at UC Irvine. This is the kickoff for our community lecture series for 2023-2024. These are organized by Dr. Brian Cummings, who's sitting in the front here. Abby Enriquez, who's down in the lobby handing out parking permits, if any of you got one of those, and really just our whole administrative team here. So a big shout out to them in terms of making these happen. I really appreciate those of you who came. We are really trying to build back our audience for this, so if you enjoy it, there's a YouTube you can share with your friends, people who might be interested, but also we really encourage people to come and just see the sorts of things we have here, interact with our faculty, interact with our students, and come and be with us on campus. We had a really robust attendance coming into the COVID shutdown. It's been such a disappointment the last couple of years trying to build back, because it really is fun to have everyone come onto campus and, like I said, share this time with us. So one quick announcement before I introduce at least our first speaker for this evening, and a quick word about format. The quick announcement is that October 11th, just about 10 days from now, this is National Stem Cell Awareness Day. And in the pre-COVID days, we did pretty big events in the center where we invited in high school students, grade school students sometimes. We did tours through the labs, lots of cool things to come and look at and scientists to talk to. This year we're taking a little bit of a different approach in terms of building back. We are heavily focusing on patient advocates or patient groups in the area. We have a lot of invites out. Hopefully we're going to be able to draw some community members in that way, and of course the UCI campus community. That's going to be an event in the late afternoon, 4 to 6 p.m. There'll be refreshments upstairs here, but also tours of our core facilities, of our Alpha Clinic. There'll be scientists around to be able to talk to, ask questions of. So if you're looking for something mildly interesting to do on October 11th, you should come and visit us. Second quick thing is just in terms of format this evening. Dr. Andersen, who I'll introduce in a moment, will lead off, followed by his student, our second speaker, who's a PhD candidate. I want to say Dr. Karri, but that's not really working. Almost Dr. Karri. If that's okay with everyone here, I'm going to ask you to hold your questions until both of their presentations are done. I'll grab a microphone, and please ask anything that comes to mind. Save them up. They're really good at answering things. And I'll bring a microphone to you. You can ask any questions that you might have at that time. With that, I guess the last shout out that I would make is we do have a YouTube channel for these community lectures that goes all the way back to when we kicked them off in 2017. There are really fantastic lectures in there. Some of them have thousands of views by people all over the country. So again, if you're interested in stem cell research, interested in the types of things we do here, I really encourage you to find that. It's easily detectable on our website. So last but not least, I would like to take this chance to introduce Dr. Bogi Andersen, who has graciously joined us here for this evening. Bogi is professor of biological chemistry here in the School of Medicine. He's also a really unusual beast when it comes to academic clinicians, because he's very active as a basic scientist, and in addition to his activities seeing patients and in the more standard clinical realm. For us at the Stem Cell Center, he's been really invaluable over his years interacting with us. One of those ways in particular with the relaunch of CIRM, California Institute for Regenerative Medicine, in 2020 is that Dr. Andersen stepped in with Peter Donovan, who's our founding director of the Stem Cell Research Center, to become the director of our CIRM Scholars Training Grant. And this is a training grant that provides opportunities for graduate students, for postdocs, for clinical fellows, to be able to do stem cell research, to learn new techniques, work with mentors, and be able to move on to their own careers in the future. So we are really pleased to be able to have him in that capacity and to be able to host those students here. He's even more important than that, though, because he also is the founding director of a National Institute for Health Center grant, a P30, that's focused on skin cell biology, which also recently was awarded its own separate training grant in order to help, again, postdocs and also clinical researchers gain experience in that area. We host both of those in the Stem Cell Center, which is our great pleasure because he's such a wonderful colleague to have. And with that, I will let him actually get up in here and speak and tell us about his research. So, Bogi, if you would join us.
Thank you. Thank you. Thank you, Eileen and Brian, for inviting Satya and I to talk here tonight. And thank you for coming to listen. So I'm going to give first an introduction about the circadian clock and its connection to nutrition and metabolism and how this can all relate to stem cells and regenerative medicine. And then Satya is going to talk more specifically about some of his current research in this area. All right, so what you have there on the right side is a schematic of the skin, and the outer layer of the skin is the epidermis. And this is a multi-layered epithelium, and in its basal layer here, along here, are stem cells that are very active. The epidermis, I would view as an organ that represents regeneration better than many other of our organs. Now, on the left side, you see our solar system, so you're probably wondering what's the connection between stem cells within the epidermis and the solar system, but I'm going to try to convince you that movements of the celestial bodies actually relate to function of skin cells. So the Earth is the third planet from the sun, and apparently life was able to evolve on Earth because water is in a liquid form. If you're farther away from the sun, it's frozen or it's a solid, and if it's closer, it's gas. And the Earth, of course, travels around the sun. So it makes one round in 365 days and 6 hours. And because of the tilt of the Earth, for half of that time, the northern hemisphere is closer to the sun than the southern hemisphere, and for the half of the time, the southern hemisphere is closer than the northern hemisphere, and this creates seasonal cycles. So that's one thing that I'm going to talk very briefly about, seasonal cycles. But the other cycles that we're really going to be talking about have to do with the rotation of the Earth around its own axis. So mentioning seasonal cycles gives me also a chance to kind of tell you a little bit about myself. So here in Southern California, we are so close to the equator, we don't really notice that much difference in the seasons. But I happen to be from Iceland, which is here, which touches the Arctic Circle, and there we do experience differences between winter and summer in terms of light and temperature, of course. And it turns out that many things in nature are linked to this yearly cycles of season. So this is one example, which is the Arctic fox, which is in the winter, has a very thick white coat. And then in the spring, it will shed that coat and it will grow a new coat, which is thinner, shorter, and is brown. So you see, so these are pictures here taken from the northwest of Iceland in an area called Hornstrand, where the Arctic fox is protected. I mention this because this is really the reason we got into circadian clock research. One of my early students here at UC Irvine, Kevin Lin, was studying the hair cycle, the activation of stem cells that are required to produce the hair. And he found that the circadian clock genes had an expression that correlated with the hair cycle. And so we even ended up testing whether the circadian clock played a role in hair growth and found that that was indeed the fact. And that may relate to these kind of seasonal cycles because the seasonal cycle mechanism may overlap with circadian clock cycles. So, and another one of my colleagues here in the Stem Cell Center, Maxim Plikus, actually has also studied the circadian clock in hair growth and is very active in looking at the hair follicle, as a model for regenerative medicine. Okay, so but what I really want to talk about is more the rotation of the Earth around its axis. So that happens, of course, in 24 hours. So, but when during that time you experience very different environments. So at night it's dark and it's colder, and that's associated with sleep and fasting. And then during the day it's bright, it's warmer, and that's associated with activity and food intake. So these are tremendous differences in the environment. And it turns out that all organisms on Earth have evolved an internal clock that helps the organisms deal with these environmental changes. And the thing to realize about this clock is that it's not passive as a response to the environment. It's intrinsic and autonomous. So if you take an individual, you place them in total darkness and even temperature, they will continue to have these cycles of activity and sleep and other phenomena that happen over 24 hours. And I mentioned that, so we find these clocks in all organisms. So you find them in plants, you find them in bacteria, and of course in mammals as well. So the actual genes that are involved are different in different organisms. So it is evolved independently in many different organisms. So that indicates that having an internal circadian clock is important for survival advantage. Now, this term circadian, which really means just about a day, was coined in the 1950s, and that's before we knew anything about the molecular clock. And in the 18th century, the Marians, he observed the autonomous movement of leaves in the mimosa plant. So circadian phenomena that are autonomous have been described since the 18th century. And you may know that in 2017, the Nobel Prize was awarded in physiology to Michael Young, Michael Rochbas, and Jeffrey Hall for having worked out the molecular mechanisms of the clock in the fruit fly. And it wasn't until 1994 when the first gene in mammals was cloned that's involved in the circadian clock, and that was done by Joseph Takahashi, which actually has been our collaborator in this study and is a leader in the field. And then in the last 20 years, I would say that work has been accumulating that links the circadian clock to a variety of diseases. And if you do a survey of genes that are involved in common diseases, you find that genes that are involved in the circadian clock are enriched among those disease genes. So this is an image that was actually taken from the press release that was given with our 2017 Nobel Prize, and it just shows you some of the phenomena that happen, that change over the course of a day. So, for example, cortisol secretion, which happens usually before you wake up, it reaches a peak there. Blood pressure is highest at night, but it rises fastest in the morning. And so one of the things that people have observed in the morning is that phenomena such as myocardial infarctions, stroke, and pulmonary edema tend to be most common in the early morning. That's when you have the maximum sympathetic activity and lowest parasympathetic activity. And so overall, nearly all parts of our physiology are modulated in this way by the circadian clock. Now you might ask, how is it possible? How can a cell have a clock? And may seem very startling. And I can try to explain it to you. So while the clocks, the specifics of the clocks are different in plants, bacteria, or in mammals, the overall mechanisms is similar. So what you have is you have activators, meaning proteins that bind to gene regulatory regions and turn on other genes. So you have these activators, and they turn on other genes that then are also transcription factors in the case of mammals, and they inhibit their own activators. And this mechanism, where you activate your own inhibitor, will lead or can lead to rhythms or waves that act as a clock. So this here on the right-hand side is an experiment that Kevin Lin, my first graduate student who first worked on the circadian clock, did in skin. So BMAL1 is one of these activators here. And by the way, so this is a graph I may show you a couple of times. So this looks at mRNA expression over 2 days. So here, this light bar here is a day, and then you have a night, then you have a day, and then you have a night. And this, on this axis here, is the amount of the gene that's being expressed. So you can see that BMAL1 has this kind of wavy pattern in expression. It peaks here at the end of the night, and then it peaks again at the end of the next night. And pair 2, which turns out to be one of these repressors here, has a different expression. So that also has a wavy expression. That is what we mean by circadian expression. So that is high at the end of the day. So you can see when BMAL1, the activator, is high, pair 2, the repressor, is low. And this is based on how this mechanism is, the underlying mechanism for creating a clock within cells. Now it turns out that the central clock is in the suprachiasmatic nucleus of the hypothalamus. So the hypothalamus refers to, it's below the thalamus. So thalamus is this region here in the gray matter region in the center of the brain, where many of the motor neurons and sensory neurons project through, but it also has other functions. And below that are neurons that play a role in the neuroendocrine system, but also that's where the suprachiasmatic nucleus is, which turns out to be the central clock. And we know it's the central clock because in the 1970s, investigators made lesions in this region of rats or other species. And when you make a lesion in this region here, then the animal loses its rhythm in activity and sleep. But you can then take that same region from another animal, transplant it back, and you can restore these rhythms. And so to emphasize what I told you before, the clock is not just some passive response to the changes in environment during the day and night. It is really an intrinsic clock. So you can take the SCN out of an animal, put it in culture, and it will continue to oscillate with these periodicity of 24 hours. Now the other thing, it's called the suprachiasmatic nucleus because it sits above the optic chiasm here. So the optic chiasm the optic nerves, they connect to the retina and they go through this optic chiasm, they switch over and project here to the olfactory cortex. And this nuclei, they're sitting right on top of that. But there's an important connection there because there are neurons that detect light from the retina that go into the suprachiasmatic nucleus. And this allows light to kind of set the time. light does not drive these rhythms, but it sets the time. So if you happen to travel, let's say 8 time zones, then you're going to have what's called jet lag, and that's actually how most of us experience the power of the circadian clock. We're somewhere during the day, but we're profoundly tired and we want to sleep. But eventually, after 5 days or so, that gets corrected, and that's because you continue to receive light from the retina that gets sent signal to the suprachiasmatic nucleus just to set the time, set the phase of the clock. Okay, so the initial idea was that we just had a central clock and the rest of the body was indirectly circadian, meaning we thought that the SCN was sending signals to the other parts of the hypothalamus or to the pituitary gland, and then hormones affected other organs. But in the 1990s, people realized that even fibroblasts, individual cells taken from part of the body, had the same genes and they also had a clock. So now we realize that the circadian clock is actually present in all our organs. And what's interesting is that we have about 20,000 genes, and we express maybe half of them in each tissue at a pretty robust level. And so about 15,000, maybe 10 or 15,000 genes in each tissue we're expressing. And out of those 10 or 15,000 genes, about 1,000 to 1,500 have a robust wave pattern according to the day. So this system has a very profound effect throughout our bodies. And that's basically takes us to skin. So this was actually our contribution after we figured out that the circadian clock was involved in activation of hair follicle stem cells. We want to understand just generally what can the circadian clock do in skin. And this just tells you how hard our graduate students at UCI work, because what I'm describing to you here on the right side is an experiment done by one of my graduate students, Mikkel Geefman. So what he did was that he collected skin samples from mice every 4 hours for 50 hours. So that's quite a bit of work and difficult to do. So what we have here is 1 day, 1 night, 1 day, 1 night, and the beginning of 1 day. And Mikkel kept working the whole time. And so what he did was to isolate RNA from all these samples. And then we analyzed the expression in a time course like I'm showing you here. So this is actually showing you circadian waves of genes. So each line here is a gene. And a green means it's expressed at a low level. Red means it's expressed at a high level. So you can see how this is a wave here. This group of genes here starts to be expressed high during the day, then goes low during the night, high during the next day, low during the next night, and then starts to be high again in the next day. So we found that this kind of pattern, we found about 1,200 genes in the skin. And in this case, we had just taken, the whole skin to make this analysis. And Satya is going to be talking about a newer method where we can look at these things in individual cells. But this gave us a picture of what's really happening. That there's about 1,200 genes in the skin that do have this kind of a circadian wave pattern. And I will get back to how that connects to the stem cells in the epidermis. So before I do that, I'm going to take a little bit detour and talk about skin. So skin is maybe we could describe it as biggest organ. It's about 21 square feet. So by surface area, it's probably our biggest organ, has 300 million cells. And it's about 15% of the weight of all our organs. And the primary role of skin is to form a barrier, both to protect our internal body from bacteria and chemicals. We don't want all this stuff to seep into our body, but also to protect us from just evaporating. So it forms a very tight barrier and prevents us from evaporating. And we can see that in kids who have mutations in genes that are important for the barrier formation. They have really a tough time and are severely ill, and some of them die because of the tremendous evaporation that can happen through skin if the barrier doesn't form. And so at a histological structure, the skin has these three main layers. So here, this is the outer epithelial layer. And then we have what's called dermis, and that contains fibroblasts that they're mostly been removed here, so you can see kind of the appendages of the epidermis, the hair follicles, the sweat glands. And then the third layer is this subcutaneous fat layer under here. And those are all very important parts of skin, but our work has focused on the epidermis. And the epidermis has what we call as the basal layer, and that's where the stem cells of the epidermis reside. They are highly proliferative cells. They're dividing, they're doing hard work, and then they give rise to these cells that are above the basal layer and progressively differentiate to form this barrier. Eventually, these cells die here at the top, and they form what's called the cornified envelope. So that's when you look at your skin, you're looking at these top layer of the dead cells, the cornified layers of the skin. And one of the reasons we're interested in studying skin and skin stem cells is that because the epidermis, it can regenerate itself. So it's not like neuronal stem cells that don't do anything. If you have I'm trying to make up Eileen here, but yeah, so they don't divide very often. These cells are like graduate students working hard, dividing all the time. And so we think that by studying an organ like the epidermis, you can get insights into how to improve regeneration in other organs. And if you need an example of how powerful these epidermal stem cells is, so this is a paper that was reported in Nature, I think it was in 2017, from Italian and German scientists. So it describes the case of a 7-year-old boy who suffered from a genetic disease known as junctional epidermolysis bullosa. And the problem in this disease is, I talked about how the epidermis sits on top of the dermis, is that it's missing a protein or has a dysfunctional protein that allows the epidermis to adhere to the underlying dermis. So what happens is that the skin becomes very fragile, so just a little friction will cause the epidermis to slough off, and in this kid had lost about 60% of the skin, and he was in so much pain. He was on constant morphine, and he was fighting infections because the barrier of the skin was lost. And so what these investigators did is that they harvested some epidermal cells from a part of his skin that wasn't actually affected. They corrected the gene in a tissue culture situation, and then they expanded these corrected cells and eventually transplanted them. Now, this is a little detail. When you look at these stem cells that have been cultured from human epidermis, they form different type of clones, and one of them is called a holoclone that is most stem cell-like. And eventually, over the course of, I think it was 6 or 8 months, these corrected stem cells took over and his epidermis and basically, cured him. So it shows you, I think, the power of epidermal stem cells and the power of regenerative medicine. Okay, so now I'm going to get back to the circadian clock and our work. So, so what we found in these genes, I told you, we found 1,200 genes that have circadian waves in the skin. So we could then ask, okay, what kind of genes are these? And so the number one category were genes involved in cell proliferation, so in proliferation of stem cells. So then Mikhail, he went on, and now he started to look at the number of cells number of stem cells in the epidermis that proliferate over the time of the day. So here, this is day, and here is night, here's day, and here's night. And he noticed that actually the epidermal stem cells have this diurnal variation in their proliferation rate. So they are proliferating most highly at the end of the night and most slowly at the end of the day. Now, we were pretty excited about this, but then we started to study the literature a little bit more. We realized that other investigators had already described this phenomena going back to the first part of the 20th century even. And this phenomena where stem cells have this kind of a circadian pattern in their cell division is common and is found also in other internal epithelia. So the other thing we found, which was surprising for skin, because we don't view skin as a metabolic organ normally. It's not like the liver that plays a very, very kind of important role in overall metabolism, or the pancreas. But it turns out that many of the genes also had this kind of circadian pattern. So here, this again is looking, over 2 consecutive days. And here, the yellow denotes high expression. So you can see this gene here is peaking and then goes down, then it peaks again and then goes down. So these are, and so we found a lot of genes that are involved in metabolism. And so in collaborations in studies with Chiara Stringari and Enrico Gratton, we were also able to look at metabolism within the individual stem cells. And we found that what the circadian clock is doing is coordinating metabolism and these waves in stem cell division. So we were not the first ones to describe this phenomena by no means, but we were the first ones to show that if you obliterate the clock, if you made a mutation so the clock wouldn't function, then these diurnal rhythm in stem cell proliferation disappeared. And the proliferation was just constant over day and night, and at a higher level. Okay, it worked. Okay, good. Sorry about that. Okay, so what our findings suggest is that the circadian clock is coordinating these cycles of metabolic activity and proliferation. And so what happens with metabolism is that you create these reactive oxygen species. And reactive oxygen species can contribute to DNA damage. And so our idea, and this is just a hypothesis, is that if you have a disruption in this process, and you're creating the reactive oxygen species when the DNA is most sensitive to damage, then over the long term, circadian disruption might lead to a stem cell dysfunction, and that could lead to aging. And so this of course is a big problem, and we know that skin aging is contributed by a stem cell dysfunction. So somehow, as we age, our stem cells really lose their regenerative ability. And this has been shown, including by my colleague here in the Stem Cell Center, Xing Dai, but this has also, of course, been shown in other organs. And so that's something that we are really interested in pursuing in these studies. And this image here on the right, I actually lifted from the internet, and supposedly it shows a truck driver who drove with his window open, and it shows you the power of UV light, which is also, of course, a major contributor to skin aging, which is sun exposure here. And I just want to quickly mention, so in addition to aging, Mikhail Gaefman, my graduate student, he showed that the time of day of UV exposure is important. So you can get more DNA damage if you expose mice at night than during the day. So skin cancer formation may be controlled by the circadian clock. Now, wound healing by others has been shown to be the wounds heal faster if they occur during the day versus night. And my graduate student, Elise Greenberg, who worked on the viral response in the skin, the diurnal viral response, also showed in psoriasis that the circadian clock is disrupted in psoriasis. And so at the end, I just want to talk a little bit about circadian misalignment and what its causes are and what could be the consequences. So one form of circadian alignment or misalignment, is that I mentioned we have a central clock and we have peripheral clocks. And the central clock kind of coordinates the peripheral clocks in the peripheral tissues. But sometimes these two clocks are out of phase in your body, and that is one form of circadian misalignment. Another form of circadian misalignment is just if your internal clock is out of alignment with the actual wall clock. None of this is good for the physiology. And the causes are of circadian mislight if you are exposed to light at night. That's one thing. Sleep during the daytime, or you take food at night. So Satya is going to be talking about the effect of food, and that is important as well. So what causes circadian misalignment? So one cause is, of course, shift work, and a big part of the workforce now works in shifts. Jet lag, social jet lag, sleep disorders, and the excessive light exposure at night. So prior to the 20th century, at night you just had darkness. But with the invention of the light bulb, things really drastically changed. And this is just, if you think about the evolution of life, this is just at the very end. And this is, shows how light pollution has increased in the United States, or projected to have increased. So there's a tremendous amount of light exposure. And people who do shift work are at increased risk for obesity, metabolic diseases, cardiovascular disease, cancer, and stroke. And so we are interested in this new field of medicine called circadian medicine, and which, the goal of this is to integrate the relevance of biological time into both preclinical studies, into clinical research, and into clinical practice. So one way we already practice circadian medicine is that I'm a clinically an endocrinologist, so I know that certain hormones need to be measured early in the morning to be meaningful. But that may apply to many other things as well. We also already know for certain drugs that they're more effective if they're applied, if they are taken at night versus the day, or vice versa. This has not been studied very much in regenerative medicine, cellular therapy, and other things, but this could also apply to regenerative medicine. And then this field is also, trying to apply behavioral approaches to optimize the daily rhythms for health, and also trying to develop drugs that may modulate the circadian clock and have effects that way. All right, so I'm going to end here, and I'm going to hand it over to my graduate student Satya, and he's going to describe his work, and some of that he's done in collaboration with two of my other graduate students, Junior Duan and Julie Wiedemann, who actually defended her thesis today. So now she's actually Dr. Julie Wiedemann. So Satya, go ahead.
All right. So, I'm Satya. I'm a fourth-year PhD candidate in Dr. Andersen's lab. And after that wonderful talk, I'd like to follow that up by focusing on the clock in the context of feeding. Specifically, how feeding and fasting affect epidermal stem cells. So, as Dr. Andersen mentioned, there are 3 layers of the skin. The subcutaneous fat here in the bottom, the dermis in the middle, and the epidermis at the top. The epidermal stem cells reside in the bottom of the epidermis in this basal layer of the epidermis, and they're constantly proliferating upwards at a rate that matches the rate that you lose cells at the skin's surface. In the case of an injury, there's a careful orchestration of cells that clot the wound, bring immune cells into the wound, create a new basal layer and collagen to replace the clot, and lastly remodel the wound site into new scar tissue. Now, epidermal stem cells are critical as they proliferate into more cells at the wound's edge and then migrate inwards to make the new basal layer. The epidermal stem cells are capable of fully regenerating the epithelium, even though other parts of the skin still form scar tissue. So they're a very good model, as Dr. Andersen said, for regeneration. So studying the regeneration here shows us that the skin is able to support a powerful population of cells. And we want to see how that works under duress, under fasted conditions. Now, speaking about the clock again, the suprachiasmatic nucleus synchronizes the peripheral clocks, thereby exerting rhythmic expression of metabolism genes, right? But the peripheral clocks are still affected by food intake, allowing for asynchrony between the clocks and metabolic organs and the SCN. Prior research in our lab found that altering feeding time can actually alter the circadian clock in skin, contributing to the skin aging and cancer, as previously mentioned. Moving forward, we were interested in the early changes by food intake itself, rather than the effect of feeding time. So, preserving epidermal stem cells are prioritized when the skin is stressed, and the food intake is a key regulator for skin. Throughout skin as a whole, as we've measured, we've seen nearly 2,000 genes change in response to feeding. Now, I'm especially interested in how the skin shifts priorities to ensure that the epidermal stem cell functions when feeding or fasting. So, our experiment will compare feeding and fasting mice, and after an extended fasting period, our feeding groups were given food while fasting groups continued to fast. We measured gene expression before and after feeding periods. So, what is gene expression measured by? We usually measure gene expression using RNA, and to understand that, we start with the overall process of DNA to protein. In the cell, DNA contains the instructions to build proteins that carry out the work in the cell. RNA is an intermediate molecule in that process. The RNA is a temporary molecule that only carries a small portion of the information from the DNA, and that information is then translated into the final product, our proteins. In the cell, RNA correlates to proteins, and we have a lot of good ways to measure RNA. And in fact, we employ a new technique called single-cell RNA sequencing, and that lets us measure the RNA in each cell in a certain tissue. We would be interested in that because in older techniques, we measured the expression in entire tissues. We ground up entire organs and then extracted the RNA for measurement. Those tests could identify differences in gene expression for the entire tissue, but the expression of different types of cells within a complex tissue couldn't be determined. In single-cell RNA sequencing, we separate a complex tissue, skin in our case, into individual cells. We can then measure the expression of RNA in every single cell. So, we can take the gene expression of each cell and then project it as seen on the right side here. In the projection, each dot is a single cell, and different cell types, indicated by color, are grouped together based on gene expression. In this case, we have that skin diagram, that basal epidermal stem cell layer here would correlate to the IFE basal group here, while the rest of the epidermis would correlate to the IFE differentiated layer here. And these hair follicles that you see are actually correlating to this UHF basal, upper hair follicle basal, upper hair follicle superbasal, and hair germ. And there's, of course, other structures that are even there, such as these sebaceous glands next to the hair follicle, and this green group right here. So this data does require significant computational expertise, and our two wonderful computational students, Junyan Duan and Dr. Julie Wiedmann, were absolutely critical in this. They were able to analyze, project, and annotate the data so that I could understand the mechanisms that are immediately responsive to food intake. So let's talk about metabolism in general first. Overall, metabolism is a complex system that can be generally divided into building tissue and energy storage or breaking them down. After eating, food is taken in and carbs turn into glucose, which is stored as glycogen. Fats are stored into adipose, and amino acids and protein are stored in cells and build up muscle. Upon fasting, the body needs to provide energy to key organs. As the body shifts from the full to the fasted state, we need to break down certain structures in the body. About 4 hours into fasting, when the glucose from food actually runs low in your blood, the glycogen starts breaking down in the liver to maintain blood glucose. After 2 days of fasting, adipose and fat are then broken down into fatty acids and converted into ketone bodies for energy. During the majority of the fasting state, full weeks after those first 2 days, our body is primarily using the ketone bodies from fat as a primary energy source. At a similar time, even muscles and protein are broken down for energy and amino acids, leading to reduced muscle mass. As starvation really sets in the body, it starts eating into its own tissues in a process called autophagy, we'll go into that again later, to continue supplying the body with nutrients. Fat, muscle, and liver lose mass early on, but other organs have to be spared from these breakdown effects, particularly the brain, kidneys, skin, and stem cells. So, in the extended fasting phase, again, we do rely on our fat storage to fuel the body, and the cornerstone of that process is ketogenesis, which turns fat into usable energy. The fat is first broken down into fatty acids, where they undergo ketogenesis in the liver. Then these ketones travel through the blood into other organs that need the energy. The ketones in our body, of course, include acetone, acetoacetic acid, and most prominently, beta-hydroxybutyrate, shown here. So, the primary enzyme for that process, ketogenesis, is actually called HMGCS2, and it drives the production of ketones in the liver. Why am I bringing up this gene? Well, given its role in the liver, we're surprised that it actually shows up as an active gene in skin as well. In the projection, we can show the expression of individual genes in each cell. The darker blues that you see here indicate that more of the gene is being expressed in those cells. We see here that HMGCS2, the ketogenesis gene, is expressed more in this group, which correlates to the epidermis and hair follicles here. Between feeding and fasting, we see that the skin of fasting mice specifically is expressing this HMGCS2 in those regions. And we want to validate that by actually measuring the RNA in a section of skin using a process called molecular cartography. So similar to the sequencing results, we actually do see that the outside of these large follicles here are expressing more of the gene in fasted skin, whereas in fed skin it's really not expressed at all, as well as in the epidermis. So this is of course interesting, as the liver does make the ketones to ship to other parts of the body, but our data suggests that ketone bodies are actually being made right in the epidermis and the hair follicles, where the subcutaneous fat locally is broken down into fatty acids and then used to make those ketone bodies in the epidermis and hair follicle. Our preliminary experiments have suggested that the skin is capable of making ketones in this way, and these ketones may be used to support epidermal stem cells and make sure that they're able to maintain regeneration and repair of the epidermis. We've had some preliminary experiments that even show that the regeneration and barrier repair is even better in fasting conditions compared to feeding conditions. However, the best way to see if this gene is really responsible for these effects is by knocking the gene out in a mouse model, which we have, and then measuring the difference in ketogenesis and epidermal regeneration. So, that brings us, in addition to ketogenesis, to autophagy. And in starvation, it triggers autophagy in the body, literally self-eating, to allow the body to obtain more nutrients from breaking down itself. Muscle and bone are broken down for amino acids, while fat from adipose in the liver are broken down for energy. However, valuable organs do need to be spared from autophagy in excess in order to preserve our bodily function. In this case, the gene TXNIP is a regulator that increases autophagy when expressed. In our data, we do note that TXNIP increases in most cell types in the fasting condition, denoted by this darker purple compared to feeding. But there is one set of cells that doesn't express TXNIP, and that's those epidermal stem cells that we saw at the bottom. This would suggest while most of the skin is subject to autophagy, while fasting, the stem cells in the epidermis are actually spared. Again, we would like to validate that. We used a section of skin, stained that skin with an antibody, highlighting TXNIP as red and the nuclei of cells as blue. In the image here, we can see that there's the epidermis and hair follicles. And in the mouse, the epidermis is particularly thin, only showing a few layers, but it's clear that the basal cells of the epidermis are not expressing TXNIP protein. Similar to HMGCS2, the best way to understand the effect of this gene is by knocking it out in a mouse and measuring the capacity of the skin to regenerate. So overall, our research does suggest two ideas regarding epidermal stem cells. The epidermis makes local ketones for regeneration during starvation, and a key regulator of autophagy is not expressed in epidermal stem cells. Ketone bodies are an important energy source for the epidermal stem cells, and that supports barrier maintenance and regeneration. On the other hand, stem cells do need to be maintained during fasting, and exclusion from autophagy supports the population of cells and allows them to live. Overall, these genes are the first targets we've found from a massive data set that are the key in keeping these valuable cells alive. Thank you.
All right, so thanks to both of you for a lot of information. Can I invite any questions from the audience? Excellent.
Interesting talk. I was curious if you've got a keto diet, does it override the skin ketogenesis? And as a follow-up, does it affect wound healing, for example?
And what is a keto diet for those of us who haven't followed?
Who don't want to be on keto.
Keto diet is very prominent, and that's actually what brings ketogenesis to the forefront of the public eye. Keto diets mimic forced ketogenesis essentially by having a lot of fats as your primary food intake, and keto diets actually do simulate fasting, in fact, and assuming in that case that it simulated fasting, it would still allow for ketogenesis in the skin. So that process would probably still occur under a keto diet.
I think that's a fantastic question. So calorie deprivation is one of the few things that we think counteracts aging, right? And that its effects may relate to increased ketones. So you're suggesting, I think, a great experiment, which is to look at the effect of keto diet on stem cell function in general. I think that's a fantastic question.
Did you knock out the in the whole body or just in epidermis?
So for TXNIP, we're knocking it out in the epidermis specifically, so that we can really look at the effect in these epidermal stem cells and the epidermis as a whole. Yeah. We did the same with the HMGCS2 as well, knocking it out in the epidermis only.
So in the when you guys were talking about shift work, for example, and their the circadian clocks got shifted. If the peripheral clocks can shift the same and match up again with the central clock, will those people then be okay and not have the bad effects that you guys had talked about? And does that ever happen, I guess? I guess.
The idea is no, that shift work is definitely detrimental, so I think just being out of phase with the wall clock is not good, So it's the recommendation is to go early to sleep and sleep well and be awake during the day. Something we break all the time. I think so. I mentioned I'm from Iceland, so my dad, he worked in a and the way the fish, in Iceland is very seasonal. So he worked in a fish meal factory. And so for a big part of the winter, they would work on these 8-hour shifts. So now after I started to study circadian rhythm, I realized that this is the worst situation you could ever have, because you're constantly changing, you're constantly going out of phase. So it might be less detrimental if you just worked at night and slept during the day, you reversed it. But if you're constantly shifting like that, that's probably very detrimental to you.
I have a question for Dr. Andersen. That was a very enlightening talk. So on one of your slides, you showed the feedback loop between the B-mel and PER2. In which cells were those tested? And also, if you derive cells from human and culture, do the primary culture, do they lose this cyclic pattern of circadian rhythm genes?
That's what I was going to ask.
Oh, and I have one other question. It's just for my curiosity. You showed the example of Arctic fox. If you move them to Southern California, do they change their shedding pattern?
Good. Okay. Well, so where the data I showed you was from whole skin, but you could have gotten it from many other cells and tissues. So about the cultured cells. So what happens when you take cells and you put them in culture is that they become asynchronous. They probably have the clock in each individual cell, but they go out of phase. So if you look in the whole population, it looks like everything is flat. But in fact, each individual cell has a clock. But you can do stuff to synchronize them. So, and people do this all the time. You can put high doses of glucocorticoids, and that will synchronize them. And then you'll observe exactly the same rhythm in the culture cell. Or you can heat shock them or serum shock them. So there are ways of synchronizing the cells. So that's the difference between the central clock. The central clock, you can take a piece of the suprachiasmatic nucleus, and those cells, they are neurons, so they're connected to each other, so they stay synchronized. But in the peripheral tissue, skin and muscle and liver, the cells don't connect in that way, so they become asynchronous. So you need some kind of artificial way to synchronize them in culture. Arctic fox, I don't know. I think it probably would cycle the same way, but it might be altered. I don't know. I'm not a wildlife biologist, so But they probably have Arctic fox in zoos here somewhere in California. We could find out.
I just want to say kudos for keeping track of all those questions in order.
First of all, I would like to thank you both for that insightful talk. I have two questions as well. The first one is mostly regarding diets. Recently, it's become a trend to do really long fasting diets, like 44 to 50-hour fasting diets. And apparently they've shown help with like improving metabolism, resetting the circadian system in the body. But then in this research, you mentioned how long fasting and starvation can like cause autophagy. I hope I said that right. I was wondering if the increased autophagy can cause to like skin disorders, and if like it's recommended to continue long fasting even if it does like help reset metabolism. So like would the cons outweigh the benefits? And then my second question is, I'm a pharmacy technician and I work with medicine, and recently more and more different types of medications are becoming extended release or short-term release to kind of accommodate for when they should be released and like help with like the circadian rhythm. And I was wondering if with this new technology of managing when the medication taken gets released into your system or how fast it gets released, we can kind of control aging or cell deterioration and things like that in order to preserve the stem cells, or if there's any research being done in that area.
So do you want to take the first one?
So when it comes to excessive starvation, starvation is a bit of a different response than just in the initial fasting. It's interesting that we have these diets that really go into long-term fasting. When you have autophagy and that early on, really it targets some of the more damaged components and things like that. And then in the long term, you start breaking down less important tissue while the main tissues are still preserved. Regarding the beneficial effects of really extended fasting, that's probably due to a lot of different effects. So it's such a complex network of genes that are being regulated and transcribed. We didn't even go into it, but things related to immune cell function and immune cell infiltration to change how inflammation works under the fasting diet. There's so many things that are regarding that when you're fasting. And there's so many different ways you can fast as well, whether you just restrict calories or if you fast for, the extended long period of time. When you start undergoing autophagy, though, that's really a breakdown of less important tissues. So you probably wouldn't see really detrimental effects of autophagy until a much longer time course, much, much further down the line. But again, those really important tissues have to be preserved from autophagy to ensure that in some way they don't get just eaten up, basically.
So autophagy in general is a beneficial thing, right? I mean, it kind of cleans up bad things in your cells. So I mean, there's a lot of fat about different types of fasting. I mean, but there aren't really studies, to support. I don't know of good studies that would really support extreme fasting and things like that. But there's a lot of evidence that calorie deprivation will extend lifespan in many different species, and so that seems highly likely. But it's probably very complex mechanisms. As to I think so. The whole idea about circadian medicine, that it makes a difference when you take different medicine, and it has been shown for some common medicines, like some blood pressure medicines, that they're more effective when you take them at night than during the day. But eventually, for this to be like a field of medicine, you need to do studies and do comparisons. And then so the idea that you can have medicine that modulates the clock, there's definitely is being pursued. So one thing is that it seems that if you have a functional clock, it will kind of prevent cells from dividing really fast. And so people have observed in cancer cells that they suspend the clock, and that may be to allow them to grow uncontrolled. So one idea is that if you had something that restored the clock in cancer cells, that you can actually slow their malignancy down. So that's one idea, yeah. But, it's unlikely that we can give you a medicine to correct a circadian misalignment. I mean, there's so many things going on, yeah.
If I may. So science communication is a big thing for me, and helping people understand that like one study does not make dogma, right? So because we've had a lot of questions just on diet and how it pertains to your work, I think there's some interesting background studies, right? And some newer data, not my field, I just peruse it casually, that are really interesting, I think. So some of the original caloric restriction studies, kind of bridging over to your question, where everybody thought, well, if you just eat less, you'll live longer and that'll be good, right? But the interesting thing that was uncontrolled in those studies is what got lockstep was the time of day that you were feeding the animals. And so it turns out in follow-up studies, because this is what we do in science, we ask the question again a different way. In follow-up studies, it may not be the caloric restriction, like eating less, that matters so much. It may be when you eat. So if you feed animals the same number of calories, but you just compress when they eat to the daylight hours, then that has the same effect as overall reducing calories, right? And so I'm sure that it comes back to your circadian clock, and this is what we do, right? You ask one question, you have to ask the next one and the next one and the next one, refine it. And so I think we're not at the point yet where certainly anyone would advise really long restriction diets, right? Or making radical changes in terms of any kind of diet, because the effects at the end of the day on stem cell populations, on autophagy, on processes that are important for your normal metabolic function are just not known, right? Mm-hmm. So avoid fads of all kinds.
Yeah. Good point. Very good point. Yeah. Yeah. I mean, there are studies that show, in terms of high-fat diet, depending on what time of the day it's taken, will affect the weight gain. So timing is important as well as calorie amount. Yeah.
So when's the best time we have cookies?
Middle of the day, probably, if you want to eat cookies.
This has been a very one side of the room conversation. Anybody over there want to join the fray? There's my name.
So I remember in one of the earlier slides, you had some data you'd implied that there was some data that the suspension of the central clock or the clock resulted in an increase in proliferation across the board or at any time point for the stem cell population. Do you think that has implications in kind of manufacturing of stem cell therapies?
Yeah, it might. Maybe I don't know if it's been tested so well, but it might allow you to have faster proliferation. If you needed to expand cells quickly, if you could suspend the clock, it might be. I don't know. It is potential. But the experiments I was referring to is if we mutate the clock and we look at the epidermal stem cells, this is what happens. And that could be controlled by so many things, so I don't know. It'd be hard to translate that directly to like cells in culture if you were expanding stem cells. And I should say about the circadian clock system, so the key genes, if you have a mutation in them, the embryo will develop totally normally, so that they don't seem to be important for embryogenesis and development of the fetus. And the clock becomes active, after birth, basically. But having a mutation in these proteins is so if you heard about morning larks and night owls, and the most extreme versions of those, they do have mutations in these clock genes. So yeah, and being a night owl is associated with metabolic disease, obesity, and other things like that. So, and I mentioned one disease that's really tightly linked to circadian clock, of course, is depression. Because a very profound effect in depression is like loss of sleep, and circadian abnormalities.
Any other takers? No burning questions. All right. Well, I want to thank you both for a wonderful presentation, wonderful set of presentations that obviously generated a lot of interest. And again, thank you so much to all of you for attending this evening. Have a good rest of your night.